The question we were left with in our last podcast was, how do you stop all of this chemical signaling that's occurring from our neurotransmitters? Remember, we had 300,000 vesicles of acetylcholine, each containing 10,000 molecules. That is a drastic amount of acetylcholine that's released into the synaptic cleft. This is to create a tremendously steep gradient, so diffusion happens rapidly across that 20 nanometer space, so that then that chemical signal can be transduced by the receiving cell. But now we have a problem. With all of these neurotransmitters in the synaptic cleft, how do we ever stop the signal? And the way that cells simply do that is by destroying the signal practically as fast as it gets there. And in the case of our neuromuscular junctions with skeletal muscle, that occurs because of a plasma membrane enzyme on the surface of the muscle cell called acetylcholine esterase. Now here you can see in our illustration, acetylcholine esterase is destroying the signal as rapidly as acetylcholine is making its way across the membrane. Now that almost seems anti-intuitive. Why are you destroying the signal you're trying to detect? Well, you have such a vast amount of signal so that you rapidly signal across the space, but you also have enzymes, so almost as the instant the signal is transduced, it can be stopped, it can be destroyed, because there may be a second signal, a second action potential making its way down the axon. And you have to stop the signal and reset so you can have the capability of having a second action potential or subsequent action potentials repeatedly and quickly. So once you break down acetylcholine to acetate and choline, it's taken up into the presynaptic cell, recycled, so you can make more acetylcholine. Now as we look at synapses and we begin to think about neuron to neuron synapses in a similar way we've seen with our skeletal muscle. Remember, with our nicotinic receptor, which is an ion channel, it allows for an excitatory postsynaptic potential. We first refer to this as depolarization because we're making our plasma membrane more positive than -70. But recall, we're using depolarization, repolarization, hyperpolarization almost exclusively for an action potential. So now in synapses such as neuron to neuron synapses, when we cause a more positive movement of our membrane potential, we're going to refer to that as an EPSP. Likewise, if we make the membrane more negative, like in axoaxonic synapses, which are almost exclusively lowering the membrane potential, we call those inhibitory postsynaptic potentials. We use those designations because the dendrites, the cell bodies, they don't have sodium voltage gated channels. And so they cannot, in fact, propagate an action potential. They can't initiate it. That's only initiated where we have a high concentration of sodium voltage gated channels, and that's in the beginning of the axon where it's connected to the cell body, in the axon hillock. In this illustration, we can see across time, we have a number of these synapses that yield excitatory postsynaptic potentials. The first two could not change our membrane potential up to -55 millivolts. I know the illustration says 50, but ignore that. Just in your mind, make that -55. And notice that they didn't just maintain the potential, but they were reset back to -70, back to our resting membrane potential. And that's because of the action of the sodium-potassium pump constantly working to reset the potential if we do not achieve an action potential. But notice on the third excitatory postsynaptic potential, we do reach threshold. We do have that action potential designated by our upward depolarization, downward repolarization. And if you also notice, that is happening at that connection between the axon and the cell body. That is where we have the axon hillock.
When we look at synapses that happen, whether it's synapses between neurons or whether we have neuromuscular junctions with muscles or other effectors, We are going to convert the electrical signal that is an action potential into chemical messages that are interpreted by the effector cell or the postsynaptic cell. And one of these chemical messengers can be acetylcholine, and we've talked about acetylcholine already quite a bit, and we talked about its receptor types. Acetylcholine and neurons that secrete acetylcholine are referred to as cholinergic. And in many instances, cholinergic neurons are going to be excitatory neurons. Not necessarily exclusively, but almost always. And that's why we use the word typically. That's how we get around in saying always or never. But typically, cholinergic neurons are going to be excitatory. Another group of neurotransmitters are going to be the catecholamines. This is a group of neurotransmitters that are derived from an amino acid. And in this case, the amino acid is tyrosine. And so you'll often hear catecholamines referred to as monoamines. What does mono mean? One. So this is one amino acid. And in this case, that one single amino acid is tyrosine. I already mentioned adrenaline, epinephrine already, to get you excited about the exam coming up. This is mostly a hormone. It's going to be a minor neurotransmitter. There may be some neurons that secrete it, but mostly epinephrine is going to come from what tissue in your body? The adrenal gland, and that's why we call it adrenaline. Your adrenal glands produce it. And as we look in the next chapter, which is chapter 12 is muscle, the chapter after that is, see, I'm like 2 chapters ahead as I have to read this stuff and get ready. On the autonomic nervous system. I forgot what number it is, it's not important. But as we look at the autonomic nervous system, we're going to see that the cells and the tissues that produce adrenaline, epinephrine, they function much like a postsynaptic neuron in the autonomic nervous system. And so that's why we include that here. But it certainly is a monoamine. Now, related to epinephrine is norepinephrine, another monoamine, a monoamine of tyrosine. This is produced by neurons. And these are used by sympathetic neurons, and we'll become much more familiar with that terminology when we talk about the autonomic nervous system. And the norepinephrine is going to, these neurons are going to synapse with smooth muscle, cardiac muscle, and various glands. Yet another monoamine in the catecholamine group is the monoamine dopamine. Now, when we look at the acetylcholine producing neurons, we call them cholinergic. When we talk about, and we'll talk about this in the autonomic section, the neurons that produce norepinephrine, we're going to call those types of neurons adrenergic, because it's very similar to the epinephrine produced in the adrenal gland, so adrenergic. The dopamine producing neurons, we call them dopaminergic. How many of you have ever seen the movie Awakenings with Robin Williams, where he's the doctor and he's trying to treat the guy that is just catatonic almost? What's the guy? Is it De Niro? Is it Robert De Niro? Yeah, Robert De Niro. And this is, I think, set back in the 50s or 60s. Well, he decides that, maybe this catatonic state is brought on by some state of the brain. And if he could just modulate that state of the brain, well, maybe, he could help. And the drug, the experimental drug that he was giving to the patients, another doctor was giving, he thought he would try it, was L-Dopa, which the body can convert into dopamine. And this is what happens in Parkinson's disease. What's a characteristic of someone with Parkinson's disease? tremors. That's because the dopaminergic neurons in the nervous system, particularly in the area of the brain called the basal nuclei, no longer can produce dopamine. They're degenerating. And you lose the regulation and the coordination of muscular movement. And so by giving patients L-Dopa, you can replace that. And there are other studies that are trying to replace neurons with stem cells and other such things that in the lab they can be marginally effective, but I'm not sure that we'll see a lot of this in the clinical setting for a long time. But dopamine is responsible for that, motor control, and to a limited extent, emotional reward. But dopamine is what is missing largely in patients with Parkinson's disease. Now, here we have an example of a synapse. And in this case, we're going to see that we have tyrosine, that's our monoamine, that we're going to modify. And in this case, it goes to DOPA, or the synthetic version is L-Dopa, which is converted to dopamine. But we can also convert that into norepinephrine. This is the synthetic pathway. Well, we've got calcium coming in, that same effect. That's going to cause the Dox vesicles to be released. We have receptors on the postsynaptic membrane. That's going to lead to chemically regulated gates opening, sodium and potassium flooding in. That's going to influence neighboring voltage gates. Boom, we've got a new action potential. Now, depending on the type of the receptor and the type of tissue, that's largely going to dictate the effect that we get. And in the sympathetic nervous system, that's going to be specific for what organ these particular receptors are a part of. Now, does this look familiar? That signaling pathway, these G proteins, you can think of them as shuttle pilots or taxi cab drivers or messengers on bicycles in New York. When you get norepinephrine binding to its receptor, it dissociates the G protein complex, and that G protein complex then goes and signals, oh, wait a minute, that's not an ion channel. Didn't the muscarinic acetylcholine receptors was an ion channel, right? We have yet another step in the signaling cascade, and this is a receptor that's called adenylate cyclase. Adenylate cyclase is going to take the energy from ATP, and it is going to use ATP and convert it into cyclic AMP, adenosine monophosphate. This is a very active molecule that is going to activate protein kinases. Remember, protein kinases add phosphate groups? This protein kinase is going to add a phosphate group to, here is our ion channel, and that is what's going to regulate. It's not a direct G protein signaling to the ion channel, but the G proteins activate the production of cyclic AMP. And it's the cyclic AMP that's important in activating the ion channels. Now that's norepinephrine. We'll talk much more about norepinephrine and its functioning when we get to that sympathetic nervous system, part of the autonomic. Serotonin is another type of neurotransmitter. But serotonin is not a derivative of tyrosine. It is a monoamine, but it's a monoamine of tryptophan. Where have we heard tryptophan before? Turkey. Yeah, you don't want to eat too much turkey because you'll get real sleepy, right? That's never happened to me. I just get real full when I eat too much turkey. But serotonin is involved in mood, behavior. Oop, maybe turkey has something to do with that there with our appetite. But serotonin, a monoamine responsible for many of these. Now, depending on the neuron, depending on the receptor, those monoamines can largely be excitatory. When we look at the autonomic nervous system, norepinephrine in many instances is inhibitory. But certainly one of the major inhibitory neurotransmitters is the GABA producing. GABA is gamma aminobutyric acid. And the neurons that produce GABA are referred to as GABAergic neurons. GABA is going to be one of the most prevalent neurotransmitters in the brain, but it leads to these inhibitory postsynaptic potentials. Now, if it's important in the brain, but it's inhibitory, how do you get anything done? Why would you not want excitatory neurotransmitters in the brain? Why do you have inhibitory? What do you think? Regulation, certainly, but look, give me a little more than that. You remember way back on the first day or the second day we talked about the thermostat? Yeah. What is the best way to really narrow the range, to keep our comfort zone very narrow? Is it just to have the air conditioning coming on and off? But if you have the air condition and the furnace, so if it gets too hot, turn the air conditioner on. If it gets slightly too cold, turn the furnace on. Well, in this case, in the brain, we have so many neurons that are excitatory, this is going to be like the air conditioner. It's going to kind of lower the temperature. And so you have a much more narrow range. But actually what the inhibitory messengers are going to do is give you a much greater level of control. And when we start to put this all together, we'll see how that really works, and works very effectively. So this is what we're talking about, this integration. And we'll talk more about synaptic integration. But here we have axosomatic synapses. And remember, we said these are either excitatory. If they're excitatory, probably acetylcholine. So you create an excitatory potential. But is this an action potential yet? Everybody go like this. No. It's excitatory, but it has to be graded. There's no threshold. There's no refractory period. Here we have a GABAergic neuron coming in and say, okay, I know you're getting that signal, and it's really going to make you mad, but don't be so mad. So it grades the potential lower. It hyperpolarizes. Now, if you have an equally excitatory and an equally inhibitory signal, what's going to happen to the two? They're just going to nullify each other, and you'll stay at that same potential. But if you have a greater excitatory potential or a greater number, then it can overcome that negative signal. And once you reach the axon hillock, if this is strong enough, then you can get your action potential. But this is like two houses of Congress, checks and balances. Before you can overcome, before you can have the excitatory signal passed on, you got to make sure that the other party thinks it's okay. You see the checks and balances there of the excitatory, the inhibitory, the greater level of control. Now, this is just showing the glutamic acid. So GABA is, in fact, a monoamine, but instead of tyrosine or tryptophan, GABA is a monoamine of glutamic acid. And so GABA, in its current form, you can see some of the pharmaceuticals that are trying to replace GABA, because it's used very, very often in treatments for depression. It certainly is an antidepressant, many of these. Now, when we were talking about acetylcholine, we said as long as acetylcholine stayed in that synaptic cleft area, what was going to continue to happen? As long as acetylcholine was in the synaptic cleft, it could bind to the acetylcholine receptors on the postsynaptic cell, and you would get transmission. You would get the transformation of that signal from the chemical into an action potential. How did you stop that? Remember there was an enzyme on the postsynaptic membrane, acetylcholine esterase. What did it do? It broke down acetylcholine into acetate and choline. Well, the monoamines don't work that way. They don't rely on enzymes in that space to destroy the neurotransmitter. They do most of their inactivation by taking the neurotransmitter back up into the presynaptic cell. Up to 85% of the neurotransmitter that is released is quickly taken back up. That effectively lowers the concentration. If you lower the concentration, you lower or lessen the chance of that neurotransmitter finding its receptor. Now, acetylcholine esterase, remember, was in the membrane of the postsynaptic cell. Here we have an enzyme that's called monoamine oxidase, but it's in the presynaptic cell membrane. And that's going to inactivate a small part. But once we get to the postsynaptic cell, there is an enzyme that will inactivate that. It's called catechol-methyltransferase, or COMT, but it's only going to inactivate about 15%. Most of the inactivation comes from the reuptake. Does that seem like a good idea? Which is a better idea? The way acetylcholine is broken down? Or the way this happens? They both work, right? If you were designing a business and you were doing the neurotransmitter signaling off, which would you rather do? What is this? Recycling. You don't have to use energy to remake your neurotransmitter. Just reuse it. Because you're only breaking down 15%, a little more than. Prozac, Zoloft, Paxil, ever heard of these guys? These are pharmaceuticals that inhibit the reuptake of serotonin. Remember we said serotonin affected mood and behavior? So one way you can continue to have serotonin stimulate your system is to keep it from being reuptaken into that membrane. And if it's in that synaptic cleft longer, it's going to have more action and have more activity. MAO inhibitors. You probably hear these on the news a lot, right? Or on television. You're watching the commercial, take brand X. We're going to do this. And people frolicking through the woods and they're living all happy. Do not take, if you are taking blah, 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 MAO inhibitors, such and such. That's where you probably heard it, right? These are monoamine oxidase inhibitors, the enzymes that break down many of these monoamines. And again, they're used to treat depression, much like the Zoloft and the Paxil, that are uptake inhibitors. But these MAO inhibitors also are treating Parkinson's disease. How would an MAO inhibitor, how would it help someone with Parkinson's disease?
More of the monoamine to be available for synapse.
If you're using, or if you're making less dopamine, then why not allow the little dopamine you have to be more active by preventing its destruction? That's the rationale behind that. And in some cases, it's pretty effective. It's not a cure for Parkinson's, right? You're not going to regrow those just by taking these MAO inhibitors, but it certainly can help improve your quality of life. Have you ever heard of that before? As physicians, as dietitians, as physical therapists, your goal is to increase the quality of life for that person that you're dealing with. I've probably said this before, but you're going to hear me say it a lot. Never forget, you are treating a person, not a disease. You are trying to improve their quality of life.
So would you want to give them like synthetic L-dopa as well as a male inhibitor?
Question is, would you want to give an L-dopa as well as an MAO inhibitor? Sure. But L-dopa is not extremely effective. And as you see in the movie Awakenings, if you get a chance to watch that, I would recommend you do it. It's an entertaining movie, but it's good from a physiology standpoint as well. Your body becomes more and more sensitized to it, so it takes a greater and greater amount versus what your body can make normally. So this is just our example of reuptake of the transmitter. You can have inactivation by MAO inhibitors or here. It's also in the membrane. and then we have the inactivation by the enzymes in the postsynaptic membrane. Now, we touched on this a little bit in the previous slide, but synaptic integration, that much greater control, higher fidelity of signal transmission, the checks and balances to make sure the information being sent is in the right form, it's in the right direction, it's at the right time. It all impacts as the net result of the excitatory potentials and the inhibitory potentials and what reaches the axon hillock. This illustration refers to it as the trigger zone. So you can have all of these excitatory potentials adding up. You've got one inhibitory. By the time all of these graded potentials get to this zone where you have the voltage-gated channels, it passes that threshold of negative 55. Boom. Action potential. The signal goes. If you converted these three to negatives and you have one positive, is that signal going anywhere? No. You have hundreds of thousands of millions of these excitatory or inhibitory synapses happening on a neuron. All of this information is coming from a lot of different sources. And it's the convergence of this information that will result in the transmission or lack of transmission of that signal to an effector. This is just a diagram showing that you've got two cells coming in. One cell gives you an excitatory potential alone. If you grade them together, boom, threshold, action potential starts. Here you have an inhibitory and an excitatory. You hyperpolarize first, and even though this excitatory potential would be enough to cause the threshold if it was by itself, you see how because of the timing of when they hit and because you hyperpolarize, you don't get an action potential. Again, just a couple of different examples of integration and how that can impact the transmission of information.